Hello and welcome back to Biology for Beginners. As always, I'm your host, Morgan. This video is going to be the conclusion of Chapter 12 and the conclusion of Unit 2. A little bit of a reminder, if what we talk about here seems a little heavy and hard to digest to you, don't worry, it is. Again, this chapter is full of a lot of stuff that honestly would be omitted from a normal entry-level bio class, so we're kind of just glossing over things here in this video. I'm not going to get into a whole lot of detail. I'm just going to scratch the surface of the topics, so not to overwhelm you if you are trying to use these videos as a study aid. But without further ado, let's talk about genomics and bioinformatics. As always, the terms and definitions should be popping up somewhere around me, and you can pause the video at this moment to grab the vocabulary we will be using today, or you can pop on over to my Patreon page, the link is down in the description below, and grab my entire PDF version of this slideshow for 100% free. However, if you do decide to make my day and become a patron, you will get all the Bio for Beginners information and material at least a day in advance. So let's get into this. The first question of the day is, what the heck is genomics? Well, genomics is just the study of complete sets of genes, or genomes. This entire field of study was born in 1995 when a team of scientists managed to determine the entire nucleotide sequence of a bacterium responsible for causing diseases like pneumonia and meningitis. If you're looking at my slide, you can see the name of the bacterium. I just don't want to try to pronounce it, so I'm skipping over that right now. But if you Google it, the CDC website will pop up and you can learn more about it. But this is not a video about this bacteria, so we're going to move on. And since the birth of the field of genomics, we have completed genomes including thousands of bacteria, most of which are in some way relevant to the medical field. So they cause diseases, pretty much is what that means. More than 200 species of archaea. Now, archaeans are just these really weird single-cell organisms that we're not going to talk about much because for most mainstream entry-level bio stuff, not particularly relevant, but that's what they are. And hundreds of eukaryotes. So eukaryotes are things like plants, animals, and fungi. Now, a fun little tidbit, the first eukaryote to have its genome sequenced was baker's yeast. So the yeast that gives us the glorious food that is bread. And the first multicellular organism to have its genome sequenced was the roundworm. And if you look down at the chart that I've included here, you can see some other organisms that have had their genome sequenced as well and mapped and all that fun stuff. You'll hear me using those terms kind of interchangeably, mapping and sequencing genomes, because they're kind of the same thing. So don't get too stressed about that. We're not going to make much of a distinction here. And one thing that you notice is the size of the haploid genome, so half of the chromosomes. So the billions of base pairs is on that x-axis, and the number of genes encoded for in that haploid genome is kind of all over the place. In general, these smaller organisms that we see here have smaller amounts of base pairs and smaller amounts of genes. Size does not necessarily correlate to how many genes and how big the genome is. It's more of a coincidence here, so don't get too caught up on that. This chart is just a fun little visualization of some species that have had their genomes sequenced. One important project in the field of genomics is known as the Human Genome Project, or HGP. And so the HGP is basically an endeavor to map the entire human genome. And there's a few reasons we would want to do this. One, humans are a narcissistic species and we're the ones that do science, so of course we want to know our own genome. But the other reasons include things like looking into like genetic diseases and genetic diversity and things like that, and where the differences are from one human to another. But this program began in 1990 and finished in 2003. And by finish, I mean 99% of the human genome was determined with 99.999% accuracy. And the reason why it's only 99% and 99% accuracy is because there's some gaps in the genome. And nobody really knows what the implication of those gaps are. But what we do know is that the exons, or the regions of genes that code for proteins, rRNA, or tRNAs, only make up about 1.5% of our genome. And then the rest is kind of divided between these repetitive segments, introns, non-coding DNA, and other types of repetitive DNA. A couple more fun facts from the HGP is that the haploid set of 24 chromosomes from a human contains about 3 billion nucleotide base pairs and estimated to be just under 21,000 genes. So that begs the question of how can humans be so complex with just 21,000 genes? And if you think back to previous videos where we've talked about this, RNAs can be alternatively spliced, and that is the main hypothesis that explains the complexity of organisms like humans. But the HGP is not dead, even though the human genome has been mapped. Since 2007, there's now an endeavor to begin mapping multiple human genomes so that we can understand how genetic variation affects human characteristics. And this brings us to what exactly is the mapping method of choice to do this, since it took from 1990 to 2003 to map the human genome to begin with. Well, the method that's used now is known as the whole genome shotgun method. And basically what this method does is it takes the entire genome and then it'll cut it into little pieces, and then the fragments will be sequenced and then reassembled in the correct order. So then you get the genome. And initially, this was not the method used, as you can probably guess, since it took quite a while. And it was very slow and very expensive. Over those 13 years, it cost about $100 million. And the whole genome shotgun method, on the other hand, takes a few hours and costs about $1,000. So that is a big difference. And the whole genome shotgun method and all of this sequencing stuff is pretty much done using computer technology, which brings us to bioinformatics. And this is just using computing technology to process and integrate biological data. So you're taking computers, you're taking software, and you're taking all of this fun technology to just process and figure out what's going on. And the National Center for Biotechnology Information, so NCBI, actually maintains a sequence database. So sequences from these genomes and things known as GenBank that is actually used quite frequently in genomics to kind of compare organisms and all of that fun stuff. And it helps us to understand the evolutionary relationships between species. One big thing that it's being used for is to figure out the evolution of humans. So Homo sapiens, which are what you and I are, and other human species. So Homo sapiens is our specific species, but other species of humans are considered to be part of the Homo genus in general. So things like Neanderthals or Neanderthal, I think is how it's supposed to be pronounced, so please forgive me there. But bioinformatics is being used to help kind of figure out those relationships and tease apart the family tree, as it were. And genomics has been such a successful field that it's led to a new field known as proteomics, I think is how it's pronounced. Please forgive me if I'm wrong. But it's just the study of full sets of proteins that are encoded by genomes. So it's just an extension of genomics. And through these different fields, again, one of the things that's most interesting to me that we're learning is these evolutionary relationships and seeing how genomes of closely related organisms actually differ. Like the little image included in this slide, which if you would like to take a moment and look at, it's fascinating, but it's just kind of one visualization of a few different species. So you can see where there are and are not differences in those genomes. But without further ado, we have gotten through chapter 2. We have closed the book on, we've gotten through chapter 12 and closed the book on unit 2. My brain is moving a bit faster than my mouth, which is not very common for me. But again, if this chapter was a little hard to digest, don't feel bad. Most of the information here is pretty technical and wouldn't be included in an entry-level bio class. So if you've made it this far, good for you. Next time, we're going to be moving into Unit 3 and beginning an in-depth look at evolution, which is something that I'm very excited about because it is fascinating. And I hope you have a wonderful rest of your day, afternoon, evening, what have you, and I will see you all in the next one. Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane B. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.